So Peter is an associate professor of immunology at the University of Utrecht. And also he's the CEO and founder of a biotech company called Cleara.
Cleara, but nice, thanks.
Sorry.
So yeah, my addition was quite last minute, but I very much appreciate the opportunity of discussing our work on cellular senescence and how we aim to target different kinds of senescent cells. And we do have some projects in the area of neurodegeneration. So it also, for me, also started with what the previous speaker said, with the realization that DNA damage is a major driver in a lot of forms of the neurodegeneration, including mouse models of Alzheimer's disease, Parkinson's, and ALS. So for about a decade or so, I have been interested in identifying how senescent cells are, are important towards aspects of aging and cancer. So I won't be talking too much about cancer for today. Let me get a pointer. But the goal was that if you understand what are the molecular drivers of deleterious consequences of senescence, then you can develop drugs to eliminate senescent cells altogether and then mitigate these deleterious effects. So, of course, the reason why we study aging is because aging is, in essence, the driver of all forms of diseases, not all, but the majority of diseases are driven by aging, and including Alzheimer's disease and Parkinson's, and you name it. So this is something which I think gradually is becoming more and more clear, that we have to get a better understanding of the aging process as a whole. And I'm a big proponent of DNA damage being one of the main drivers that promote aging and deterioration of the cells in our body at the molecular level. So in our lab, we are focused on 4 different areas. Like I said, cancer, which I won't be talking about, metabolic diseases, musculoskeletal diseases, and also neurological disorders. And for all of these, there's ample of evidence. These are just some of the papers that we found and were able to find at the time, that senescent cells are associated with disease in humans and drivers of the disease in mice. And now there are several papers showing that senescent cell clearance in mouse models counteract signs of Parkinson's and tauopathies. Whether it's Alzheimer's or not, you can debate, but tauopathies at least. And as I'll show you in a minute, we have more, data on ALS mouse models. So about 5 years ago, and it started about 10 years ago, we identified that, and we used these mice that are very prone to DNA damage. So these are progeria mice that accumulate DNA damage at an accelerated pace, and clearly they age extremely rapidly, and they have all sorts of neurological impairments, not to say all sorts of other forms of tissue dysfunction, including hair loss. We identified at the time, we were one of the first to do this, what could keep senescent cells alive. So again, these are very damaged cells. Senescent cells are very high in DNA damage markers, but they don't die. So there must be something that prevents these very damaged cells from being killed by the body. And we identified that that's a complex of FOXP53, at least in this type of senescent, not in all types of senescent. And if we made a peptide to break this complex, we could counteract at least these signs of aging. I'm not saying it's always the case, but in these mouse models for accelerated aging based on extreme DNA damage, we could counteract the signs of some signs of aging. We now identify that there is different kinds of senescent cells. So this is actually what I think the field of senescence is moving more and more towards. And we just don't know how this looks like in neurodegeneration. So that's all very early. But if you do single cell sequencing on, in this case, in vitro cultured cells, and we have done this also in iPSC-derived neuronal cells, you see that there are different clusters of senescent cells that we can identify. And to me, this argues that we need to make drugs that are specific for these subtypes of senescence. So just to name some that we are very much interested in, these are markers that we find to often anti-correlate, even though on bulk analysis, which is what most people study senescence on, they do population analysis of senescence. And then you find all of these markers elevated, but at single cell level, they often difference, often anti-correlate. And how I got interested in senescence heterogeneity and how I think we should also consider this in neurodegeneration is we already heard from the previous speaker that interleukin-1 A and B are very important, for instance, in his models, or actually in patients as well. for activation of astrocytes towards microglia. But we also observed that this is very much true for targeting of senescent cells. So if you apply this anti-senescence compound, or FOXO4-derived peptide, we could kill 70% of all senescent cells. But if you now co-stimulate the cells with interleukin alpha and beta, actually we could kill all of them. Vice versa, if we block the I1 pathway, we could not kill. The targeting was much less efficient. So we think we have a compound that is very capable of targeting DNA damage associated forms of senescence associated with the interleukin response. And to put this all into neurodegeneration, that's my last slide actually, we identified in familial forms of ALS, the M37V mouse model, also in patients actually, that there's a strong increase in senescent cells. For instance, marked by loss of lamin B1, which is one of the more potent markers to use in vivo and in human tissues. Vice versa, if we ectopically express the mutant form of TDP43, we see that there's a strong increase in PML FIS3BP1, and vice versa. In senescent microglial cells, you see that there's an increase in TDP43 if you induce senescence by exogenous means. So there's lots of links between the familial form of ALS and cellular senescence, and this is what we are exploring in mouse models at this point. So questions that we have is, how does senescence affect neurodegeneration? We do co-cultures of iPSC-derived astrocytes, glial cells, and neurons, and also in vivo experiments in mice. We try to identify which subtypes are important and prevalent during the different kinds of neurodegeneration. Again, our main focus is ALS, but we have links to Alzheimer's disease models, although of course there's a big question whether there are any good mouse models for Alzheimer's, but that's another discussion topic. And we have a compound which is CLO41A3, which I'll be talking about on Thursday, which is an improved version of this FOXO4 peptide, which in vitro works very well against counteracting contraction of the neurons in these co-cultures. And we are now trying to use it in mice and see if we can then stall or maybe ideally reverse cognitive decline. Yeah, that's what we do. So thanks.
Wonderful.
Congratulations.
Quick question. What did you say was your marker to identify senescent cells in human brain?
We often work with a negative marker, which is the loss of the nuclear lamina, lamin B1. Of course, yeah, so of course there are many markers, right? And I think lamin B1 is one of the more potent ones. The downside is that healthy cells have varying degrees of lamin B1. So you usually also need to do a cell type marker to know for sure that you're only looking at glial cells or only looking at astrocytes. And then within that population, loss of lamin B1 is a very powerful marker for senescence.
Okay, and when we get to the group discussion, I would love to hear, Ted, have you looked at lamin B1 levels in your reactive astrocytes and microglia, and how does that relate to a senescent cell that Peter is looking at? How are those intersecting? But we can wait because
So we'll go to the next speaker with Jonathan Rothberg. Jonathan is the founder and CSO of 180 Life Sciences, but is also has been highly involved in autoimmune encephalomyositis research with Larry Steinman, and he has very exciting data to share about his soak proteins and amyloids.
So I'm simply going to try to reiterate very quickly how heat shock proteins, fibrils, and nicotine are using a common pathway of immunosuppression, and then even touch on what 180 Life Sciences is doing in this area as well. So next slide. So the story began when Larry Simon's lab was looking at what genes were overexpressed in mice with EAE, which we all know is a model for human multiple sclerosis. And among the top 3 proteins in both the protein screen and the RNA screen, alpha B crystalline was a very common gene transcript. And to test the hypothesis that we originally had that it was going to be deleterious, there was the knockout mice. We developed EAE in those animals. And interestingly, instead of becoming better, they actually exhibited more intense symptoms. And then reciprocally, we treated EAE mice with the small heat shock protein, either IP or IV, and it improved the symptoms. And then we showed the chaperone activity of the protein correlated with therapy. Next slide. We could make peptide surrogates, which were both chaperones and as therapeutics. And most importantly, we demonstrated that the peptides were therapeutic and chaperones only when they form amyloids themselves. And so we could conclude that the small heat shock proteins and the amyloids had a common pathway. And the last bullet is very important, is when we did co-immunoprecipitations, we pulled down alpha-7 nicotinic acetylcholine receptor from both B cells and macrophages. Next slide. And the work of David Eisenberg, whom I've cited, is showing that the dominant feature of the amyloid is the cross beta spine. And we were then able to reduce the peptides as surrogate amyloids to 6 amino acids as he has crystallized. And every one of those amyloids was therapeutic for EAE. Next slide. And then, of course, the pieces all fell into place when you read Kevin Tracy's work on the vagus nerve. The alpha-7 nicotinic acetylcholine receptor is central to signaling the vagus nerve and or signaling in peripheral innate cells such as macrophages and other antigen-presenting cells. Previous work I'm citing showed already that amyloid bound, as we found, to alpha-7. And of course, nicotine also is therapeutic in EAE and of course binds alpha-7. And the last point, as shown beautifully by Kevin Tracy, that the reflex control of immunity is central to this pathway. So that's the slides, and I would bring up for the purpose of 180 ELS that we're working with Mark Feldman and Larry as founders of looking at anti-TNF in fibrotic indications. But the key point that for this session is I had the opportunity to referee a paper that was using agonists of TNF receptor 2, which unlike TNF receptor 1, which is expressed in the vast majority of cells, TNF receptor 2 is principally on CD4 and CD8, Tregs, and a few of the myelin-producing cells in the brain. This paper showed that agonists of TNF receptor 2 increased cognition, limited amyloid production, and was generally therapeutic in an animal model of Alzheimer's. And I had the opportunity to write a comment saying that the rhetorical question, is inflammation, are amyloid fibrils inflammatory, or does inflammation induce amyloid? And that chicken and egg point cannot be over exaggerated because it changes entirely the pathway for therapeutics. Clearly, we are at a stage early on, similar to early on in rheumatoid arthritis, where a variety of anti-inflammatories now have been shown. And similarly, as the previous speakers, the TNF receptor 2 pathways, a tremendous opportunity, CGAS, STING, NLRP3, probably all will have therapeutics that are going to help this entire class of indications. And I will leave it there and we can bring this further up into discussion.
Well, there's clearly really interesting common themes from all of the speakers tying inflammation to neurodegeneration and identifying inflammation in various different ways as a therapeutic new target. But I think so we could discuss that more, but want to just finish off the speakers with Micah Murphy, who is, I met in our pre-session. He's a genius at designing clinical trials in this incredibly difficult space that has been sort of a wasteland of clinical trial success. We're just not having clinical trial success for neurodegeneration. And I think we have to rethink how we design these trials. And Mike is at the forefront of that effort.
Yeah.
Well, thank you very much for that nice introduction. I hope you can hear me okay.
Yes.
My particular interest is translating novel hypotheses into druggable targets with innovative experimental designs. And I thought today I would share with you 35 years of experience working on small molecules, biologics, advanced therapy medicinal products across a host of different organizations over these many years. None of the information I'm sharing is proprietary, but it is a collective experience, I think, of the field. We begin with where have we been and where is this going? So many of you might remember, particularly in Alzheimer's disease, precursor loading strategies, esterase inhibitors, muscarinic agonists, and so forth. Today, in the lower right-hand corner of this slide, you see gene therapy, stem cells, modulation of the microbiome, which is an exciting concept, and increasingly repurposing drugs, which I hope we do have a chance to talk about, that have known pharmaceutical qualities and yet allow us to test interesting mechanisms of action. Now, as innovative and as exciting this evolution has been, what is not as much appreciated is that the clinical research methodology has paralleled this development. Diagnostics, assessments, clinical pharmacology constructs, biostatistics. It is enabling us to rethink how we design and implement novel pharmacotherapies across the neurodegenerative space. Some of the things we've learned over the time is actually a process issue. So I would say in the last 5 years particularly, there's been an explosion of interest of making what I call matrix teams. Matrix teams are a diverse collection of individuals, each bring a unique talent to the table, following the old adage that no one by themselves is as good as everyone together. We come in very, very early with most of the sponsors we work with, certainly at the time of lead optimization, sometimes when there's a hit to lead. And the reason we do that is we want to understand the science, the pharmacology, the pharmaceutical properties of the agents being developed. We want to learn how to translate that again into a novel hypothesis, a druggable target, and using some innovative trial methods that maximize the value, particularly in early phase clinical research. Early phase clinical research in neurodegeneration is of seminal importance to enable an inflection point to move the asset along to later stages of development. Some of the many conversations we have, I use this slide to illustrate that, and that is the proper mix of phenotypic versus targeted-based screening. Depending on where you sit, this will be passionately debated. In central nervous systems particularly, first-in-class drugs are usually based on phenotypic screening. However, follow-on drugs tend to look at more targeted-based screenings. The problem we've had in neurodegeneration that many of you, of course, could speak to is that the disease homology is kind of plus minus. There are biochemical, histopathological, and behavioral linkages, again, plus minus. These are generally biased screens. There's certainly an inability to map into patient toxicity, such as with ARIA and VGE. But the most important issue we have is inconsistent clinical benefit or no clinical benefit, even when there is unambiguous target engagement, both preclinically as well as clinically. So the proper mix of phenotypic and target-based screening usually occupies pretty much of the discussion in the year it takes to move one of these assets into clinical development. We've also learned to look clinically at different disease phenotypes more commonly under one construct. There are more commonalities in some cases than there are differences. So do we have a nosology-based programs? What are the core phenotypic features? Is there a genotypic profile that's particularly important? Indicative and supportive clinical biomarkers, which is everything in early phase clinical research. The basic problem we have then is how do we exploit a situation where actually, if we thought about it just a bit, we're maximizing heterogeneity rather than increasing homogeneity by looking at a broader construct here. All models are wrong, but as we say, some are very, very useful. And that is a very important construct for us. As we look at this particular side, by the way, contingent on the program, there's likely, as I mentioned, to be more heterogeneity in terms of the sample. This has a direct impact on sample size calculations, power calculations, subset analyses. It's also quite probable that some of the dose exposure response relationships are not linear or monotonic. They are not linear or they do not asymptote with increasing dose. It's also equally possible that some of the more ancillary symptoms would mature at a different rate. So generally speaking, given our conversation today, the discussion will be how much heterogeneity can we bring into the program to enable us to look at common molecular targets for diseases that have a markedly divergent clinical phenotype. All of this, of course, occurs in the context. The reality of clinical development is one where both the scientific mandate and the business mandate must equally be balanced. So on the left-hand side, do biological concepts translate into pharmacological properties we could look at? Are there predictive modeling, as I previously indicated? No one model would be adequate in our opinion. Can you validate target engagement clinically? That's been occasionally a disappointing proposition. And because we're probably going to be talking about upstream targets, off-target pharmacology becomes a major limiting factor moving ahead, along with biodistribution and toxicological readouts. On the right-hand side, we also live in a world where there are business mandates. What is the available runway for a company? To design a 5-year program for a company that may have 3 years of funding is not particularly useful. Does the company have a single molecule or a platform technology that we should exploit? Or the innovative development pathways are very established ones. Having an established clinical development program is predictable and somewhat comforting. But however, having an innovator program offers prospects for insights that one had not previously considered. And for all of these organizations that work with advanced therapy medicinal products, particularly, the need to create inflection points in the development program where there's a certain generation of interest and excitement that validates the target, validates the approach, and prompts additional investment and resources. So my last slide is this one. I think in terms of what we're going to speak to today, that a portfolio of clinical trial methodology is absolutely mandatory. The ability to use different designs, to pull out different arrows in the quiver, to look at different ways of exploiting particularly early phase clinical research, and on the lower right-hand corner of this slide, to look at disease that are capable of incorporating disease subtype genetic heterogeneity, maybe the concept of master protocols such as baskets and umbrellas that our friends in oncology have used for many years have an opportunity now in neurodegeneration. So with that, I will stop presenting and appreciate this opportunity to speak to you. Okay.
Thank you so much, Mike. I think the big question here is we have speakers who have identified really promising targets, but how do you get from that really promising target to a proof of concept study, which is really difficult in a reasonable cost and period of time to show benefit in any one neurodegenerative disease, whether it's progression of ALS or improvement in Parkinson's disease symptoms or improvement in cognitive function. How do you get even just a proof of concept? And then from proof of concept, how do you hit sufficient data set to get regulatory approval, which has turned out to be so difficult given the kind of high bar that regulatory authorities have set? So would love to open that up for discussion since all of the panelists are involved in or moving towards development of drugs for neurodegeneration.
Not only high bars from a regulatory perspective, but evolving sentiments, I might add, particularly in neurodegeneration. which actually is quite exciting. You know, it's not a static motif at all. There is a receptivity to innovative concepts, trial designs, the mixture of clinical and biomarker-based assessments. Very interested in innovative study designs, particularly for early phase clinical research. You know, does it get in? Does it hang around? Does it engage target? Does it do so in a way that's reasonably safe? Those are basic foundational issues that every drug development program has. So the reason I personally endorse a very early engagement with our friends in discovery is to appreciate what the strategic program needs to look like, from pre-IND, pre-CTA enabling activity, all the way through proof of concept and eventually a registration program. I do believe it's a process and not a static concept that would work best here. One could point out many, many different issues that a clinician would need to deal with, particularly as we engage more upstream targets. But nevertheless, that's the excitement of working in neurodegeneration.
So I guess I think I understand you could give a drug, and if it did need to penetrate the BBB, you could see if it was measured in the CSF. Target engagement could be tough if it's, eliminating senescent cells in the brain. How do you show that in a CSF or a peripheral blood sample? So, you can't do brain biopsies.
Oh, she is it.
Yeah.
Usually it's a combination of fluid and electrophysiological and clinical biomarkers that give you a proxy for what you believe to be a clinical outcome. Clinical outcomes that are meaningful are very hard to do with small sample sizes and limited durations of exposure, which frankly are the majority of the sponsors that we work with. 96% of our client base are virtual and small companies who have sufficient resources to engage, and that's called a proof of concept program. So automatically that means the need to leverage the patient sample to maximize signal detection, to use a collage of different assessments, kind of a body of evidence review, if I borrowing the term from the orphan disease space, to look at the collage of measures and ask the question, what is the patient trying to tell me about this product? And then one goes on from there. So I believe that given our discussion today, it's the early phase research where the differentiation needs to occur. The later phase research tends to be more codified, although there are some exceptions in that regard. But the earlier phase research is where the innovation can really have maximum flexibility. And we are seeing that in various neurodegenerative disorders.
So
I could bring up an interesting point that I would not want to touch a Alzheimer's trial just because of the cost and difficulty. Our company, again, is looking at a much more modest indication, which is post-operational delirium. Mm-hmm. This is an interesting situation where elderly people fall over, break a hip. They are perfectly cognitive going into the operation. Post-operational, they've lost tremendous percentage of their cognition, and the poor anesthesiologists over the decades have bore the brunt of the blame. It turns out that it has to do with the operation opening the blood-brain barrier, yes, and a variety of inflammatory cells entering at the time of the operation, leading to massive loss of cognition. Mm-hmm. We have shown in animal models and are doing human clinical trials with anti-TNF as a prophylactic. You simply inject anti-TNF as if it was a rheumatoid patient prior to the operation, and we expect a vast reduction in loss of cognition, or being more positive, saying maintaining cognition. Yes. So there's an indication that I think is approachable. It's within the budget of a small company. Mm-hmm. And from there, again, you can point out the damage inflammation does to cognitive impairment.
Yes, I know this model, Jonathan. We worked extensively in this space maybe 15, 20 years ago. And the difference is whether it acutely modifies cognition, which is not the case given this scenario, or whether it's using it as a prophylactic therapy. The challenge amongst many that we had, the benefits of the model is I can control the model. I know exactly when the insult's going to occur. And it's a well-described clinical phenomenon. The disadvantage of the model for prophylactic therapy is there's a certain recovery rate that takes place. It's usually a longer-term study to actually assess that with a larger patient sample. So it's a great model, and this is when we, clinician would need to talk to you a lot more about the MOA of the product, what you're intending to demonstrate, what domain of cognition. You know, the neuropsychologists like to tell everyone, and maybe they're right on this, that the advances in neuropsychology have actually kept pace with the advances in neurobiology. So the ability to look at different domains of cognition in a very selective and sensitive way sounds like that might be part of the solution here with minimal sample size.
But I appreciate all those points. The key, again, that you're bringing up is, remember, large pharma has spent hundreds of millions of dollars with their anti-amyloid, and then all the Me Too trials, which have been a tragic waste of funds. Innovation is still in these difficult neurodegenerative diseases. It's going to take a great deal of money and time to do those trials, which is outside the scope of a lot of the small innovative groups.
Okay, so going back a bit to the basic science of the whole system, I want to go back to Ted. So Ted, could you please elaborate? So this MIF factor, does it affect also mitochondrial responses, or is it just the DNA damage response? First question.
Well, MIF is, like, it's called macrophage migration inhibitory factor. It was actually the first cytokine that was discovered. And it's an atypical cytokine. And the reason it was called migration inhibitory factor is that when it was put in cells, it actually inhibits almost all immune cells from migrating. So it's got very powerful immune functions. It was discovered to have tautomerase activity. So tautomerase inhibitors, tautomerase knock-in mice were made, but not all the effects that have been attributed to MIF were accounted for by the tautomerase knock-ins and the tautomerase inhibitors. So we came along and discovered that it has nuclease activity, and we think it accounts for some of the many other functions of MIF. But whether it feeds back to the mitochondria, we don't know yet, but we're looking at that.
Okay, and one other question is, you're probably going to shoot me for this question, but what do you think actually T cells are doing inside the brain? Do you think this is an antigen-specific response in your models, or this is just a bystander response?
Well, the data is accumulating that it's that they're actually responding, and for instance, in Parkinson's disease, Dave Sulzer with Dr. Setti at University of California, San Diego, had identified sequences within alpha-synuclein, and alpha-synuclein is the major protein that accumulates in Parkinson's disease, that T cells recognize specific antigens near a tyrosine 39, which is internal, and then also C-terminal peptides. And those activate the T cells, CD4, CD8 respond to that. And so they would argue that it's an active participant in the process, not a bystander.
Yeah, which is very interesting. So
I would love to.
One major question that you really have danced around, which I don't think has an answer, and it's not just rhetorical, is how much is inflammation systemic inflammation different from tissue inflammation. The anti-TNFs, as we all know, the IL-6 receptor blockage is working systemically. The question will be, and we do know that, as I just alluded to, treatment, as the previous speaker was saying, of external cells systemically that then penetrate the brain is one strategy. And we don't know whether that is the basis of the problem, or is the basis of the problem going to be resident macrophages, whether it's the astrocytes or the myeloid-derived cells in the brain that need to be quenched. Because those are, one requires drugs that penetrate the blood-brain barrier, and the other pathway, like the anti-TNF receptor 2 agonist, is acting systemically and then having Tregs enter the cell, enter the brain. So those are, we need to try to understand the distinction about which is going to be a more effective strategy.
So what are your cells that you think the amyloids are actually targeting? Are they just targeting the innate response and the microglia, or the T cell response as well?
Well, one thing about our theme is that amyloids are both immunosuppressive as well as immunostimulatory. So I would just take amyloids off the table. I just take them away. Pathology drove this field for 100 years. And it's quite clear that that's not the solution. It's in my eyes, I'll be dramatic enough to say it's an effect and not a cause. So that you go upstream, as the previous session emphasized. Senescence is releasing DNA within the cytoplasm, induces C gas. There's a reason that you have a fundamental foundation of inflammation in elderly brains. And that inflammation, as a matter of degree, is causing havoc. And
So
that has to be suppressed.
So that gets to my question. Is brain inflammation due to senescent cells in the brain? And again, from what I understand, there aren't very many, or is it due to activated astrocytes and microglia? And what is the intersection between those groups?
It's probably a combination of both, that, these proteins which accumulate. I mean, if you look in animal models, and if you look in cell culture, and if you look in the brain, there's clearly, and through single cell sequencing now, there's clearly activated microglia, there's activated astrocytes. And so they're responding in ways that are pathologic and deleterious to the cell. And so one, it occurs inside the brain. DNA damage is occurring, which can then be released out in the cytosol, which is then releasing, activating C gas and sting, which brings in inflammatory components. And then, as Jonathan said, in many of these diseases, there's also, the non-neuronal immune system, which is participating in the response to these degenerative diseases that are also contributing. So it's probably both. You need to inhibit what's going on in the brain. You need to dampen down what's going on in the periphery. I'm not an immunologist, but I've had immunologists tell me that the immune system is constantly sending in T cells into tissue to sense what is going on. And then those T cells sense, they may activate the local inflammatory cells there, but some of them go back out into the periphery to recruit more T cells or other cells back into where the damage is going on. So it's both, at least my understanding.
Yeah, I totally agree. And I would not, I'm a huge fan of practical experiments of a therapeutic working or not working. And what I would love to see is a spectrum of anti-inflammatories being tested in these indications and see whether or not they have benefit. And if they do, then you can support these hypotheses a little better. But as to this day, I'm not sure people know exactly what is causing inflammation in a lot of autoimmune diseases. So I wouldn't necessarily say that's central to the focus of a therapeutic. The key is to acknowledge inflammation, work on whether or not a therapeutic that, because there are now half a dozen strategies and pathways, see whether they work individually or in concert and are being more effective than reduction of the amyloid, and do a head-to-head sort of comparison. And that will be the truth that will come out in the next 5-10 years.
Yeah, I
So
would
think
just,
in mouse models this was addressed, right? So I don't know who told you that there's not a lot of senescence in neurodegeneration, because it really depends on the biomarkers. And I would like to echo what Ted mentioned, that there's definitely evidence for DNA damage and senescence in a lot of mouse models for neurodegeneration, and clearance there of the source. So you can of course apply anti-inflammatory drugs, but then you need to do this continuously, right? And that has all sorts of unwanted side effects. But if you clear senescent cells from these mouse models, they absolutely do better. So taking away the source of the problem is beneficial for sure, right? And so I think this is what is so promising about the field of senescence, because nowadays you have biomarkers for on-target engagement, for instance, lipids and also mortality scores using metabolites that can indicate if your senescence targeting drug was effective or not. And what I'm also thinking about, now that we're on the topic of on-target engagement, is the methylation clocks, right? So these are booming and aging. And we know from work from Dawson Bodegom that actually 80% of the methylation clocks can be explained by active versus naive T cells. And I'm not sure how this is in active versus naive glial cells, but I would assume it's similar. So for on-target engagement, you could probably use the methylation clocks to see if your therapy was effective, right? So I think, again, from the mouse models, anti-inflammatory drugs work, but they're not great. But removing these cells altogether is safe and absolutely beneficial in tauopathies, ALS, and Parkinsonism. And you can measure mortality clocks and methylation clocks, both. Yeah.
so quick question, what are the senescent cells? What cell type is it?
That's exactly a good point. So that is what I was trying to argue, that we should step away from that definition, which is also why I was happy with Ted, making that argument. There are different kinds of senescent cells. So what was done so far was P16. So P16 positive cells, where they can be eliminated with a suicide construct. And in mice, where you eliminate P16 positive cells, then usually the neurodegeneration is declined, right?
What are those microglia, neurons, astrocytes? I'm just curious.
Right. So it's actually not in the neurons, so it's the glial cells, right? We think so, the microglial cells. Yeah.
But have you seen? Okay, so coming back to our previous discussion that we had, that the aging immune system affects other systems, do you think that maybe some sort of decline in the immune system in the elderly affects neuroinflammation inside the brain?
Yeah, most likely. I think because immune cells, probably they secrete something to enforce the microglial activation and the senescence response, right? So yeah, to an extent it's intrinsic. So DNA damage in the microglia can, we've seen that actually, that there's a buildup of DNA damage in the microglia during aging. And I think an aged immune cells no longer removes these cells, right? So the argument is twofold. There's more buildup of DNA damage during aging, and there's less clearance of these cells during aging. As a consequence, the net result is an accumulation of senescent cells during aging.
Okay.
So it's twofold.
Yeah.
And then I want to go back to Mike. And I was fascinated it's so hard to do a neurodegeneration trial. And then I would think, now if you do a basket trial,
Yes.
you have a smaller N for each of your baskets, and then you have to show in a smaller N something compelling enough to choose which indication to pursue. You know, in cancer, it's a little easier because cancers never regress. So when you can see a cancer stop growing or getting smaller, you know you have efficacy. But I would think it would be tough to see some evidence that you were going to have clinical benefit in a basket trial of a variety of neurodegenerative diseases. So I'm fascinated how you solve that issue.
I'm not sure anyone has solved it, but I can tell you what's being discussed. So our friends in oncology have certainly paved the way for both pilot and confirmatory basket studies. So a basket program, for those not familiar with the design, is an asset looking at multiple different targets, as opposed to an umbrella, which would be multiple assets on one target. So the idea would be these diseases are linked by a common molecular mechanism, even though they have slightly different clinical phenotypes. The way that it's normally done is you have an enhanced phenotype for each subtype, then you kind of play the winner and amplify the number of patients in one subtype, one disease type as an example. One uses certain statistical techniques that allows you to make a reasonable inferences on the particular subgroup of interest. And then mostly they're exploratory in nature. So they're not confirmatory by any means. There is a technique for a confirmatory basket study, but my experience working with many companies is outside of oncology. That's not ready for prime time in neurodegeneration. We mentioned another thing that Jonathan brought up. So getting ready for the call, I had the fellows look into, so how many interventional studies are out there with neuroinflammatory hypotheses? There are 39. So if we really were very strict on the criteria, there were at least 13 that we thought were quite respectable with 11 different mechanisms of action. And interestingly, the results are kind of mixed, not negative. So one of the exercises one might entertain moving forward is to examine the studies that showed a favorable effect and try to discern, is that design or is that molecule? Is you know, sometimes I say, you know, drugs fail all the time, and occasionally it's even the drug. So we have to know, is it a design issue? Is it a hypothesis issue? But I think there's enough extant data across neurodegenerative conditions, mainly their Parkinson's, Alzheimer's, MS, quite a bit actually, but there's enough data out there that would allow one to mine the data for insights in terms of novel hypotheses. So I hope that's answered the question a bit. So I did want to fill that note in from Jonathan, because I do think there's a lot of research done, as you might know, in neuroinflammatory hypotheses of these different diseases. I don't know that we've systematically evaluated the studies to inform a discovery program. But our first baskets have been in actually non-neurological conditions. They're in psychiatry. So the design is extrapolable to many different indications with limitations.
Right. So Jonathan, a quick question. What do you think the TNF receptor, the TNF receptor 2, you're targeting actually? Is it like the Treg cells, or you're decreasing the T effectors?
Well, you don't know that. That's the other side of the coin. But it's a response that I'd make to Peter, that, because I've heard this innumerable times from other people saying anti-inflammatories are not effective. That broad statement really is of not much value. TNF receptor 2, as you know, there's no commercial agonist, because it's designed, as I was alluding to, for the receptor to be on a cell surface to bind TNF on a cell surface, which is completely different than how TNF alpha works in solution or in serum. It requires cross-linking of multiple receptors, which these clever people in Germany designed almost a Poseidon fork to be able to stimulate. Their work was a proof of concept because it's not going to be a practical drug. And it's going to be very hard for the drug industry to make a small molecule that is capable of cross-linking that receptor. But that receptor is the best stimulatory mechanism to expand Tregs that has yet been defined. And it's a tremendous opportunity to explore that situation for a variety of situations. So I think it's just an example that there's a lot more about anti-inflammatories that we have yet to discover. And Mike's point about the behavioral diseases, I'm a big fan that schizophrenia has been dramatically shown to be linked to the complement system and hyperpruning.
Yes.
And gamma interferon would be a fantastic drug to try because it downregulates virtually the entire class of complement proteins and acts as a inhibitor of microglia, which of course are involved in pruning. So
But it might
the
enhance
neurodegenerative diseases might learn from trials coming out of behavioral diseases.
Yes, yes, I agree fully. There is a lot of interest in that. We occasionally do encounter that hypothesis out there in the field. All of this speaks to there are many, many different entry points for these compounds. And so I think the innovation is how you do it, because all of the hypotheses are really fascinatingly interesting. But no one company can easily address all permutations. So I go back to how do we enable earlier phase clinical research to have an interesting and informative readout for a certain hypothesis. I think the traditional ways that we're probably doing clinical research, which are highly codified actually in the neurodegenerative space at this point, don't serve us particularly well for early phase investigation. The confirmatory studies that we're all used to reading about, those are fairly well set in stone, even those change a bit. But it's the early phase research where the differentiation occurs, where the clever experimental designs and the interesting hypotheses are explored, where innovative methods of statistics and we so many that we haven't talked about yet, but are very common in the oncology space. I often say our colleagues in oncology are 5 to 10 years ahead of us of those in neuroscience, but we shamelessly borrow from them, from many of the early phase programs that we're involved in. I think it's really exciting that we're having this multiplicity of targets and concepts. I think there's a huge armamentarium of ways to do this outside of the traditional motif that we're all familiar with. And by engaging very early in discovery efforts, that's why I'm saying prior to you even have an asset, just to understand the science better and work through the issues so that we have an informed early phase development program is really the important message, at least from my opinion, working in this field these 30 years.
Peter, can we go back to Peter?
Well, I think we have some
questions? Okay.
questions from the audience
Okay,
that I
sorry.
wanted
Yeah.
to give you. So one was progress in frontotemporal dementia research. Anyone want to comment? And the advantages of doing trials in frontotemporal dementia versus Alzheimer's disease, I think, is an interesting.
Well, certainly there's good precedent. The one that I could publicly speak to, because it's well known, is a Tall RX initiative. By full disclosure, we helped design and operationalize and analyze the study, a very elegant international study with certain learnings obtained from doing it. So there is precedent for doing really very nice clinical experimental designs with multiple different hypotheses embedded within that type of program, specific measures, specific assessments, but the general collage of clinical neuroimaging, biochemical readouts occurred in that particular study, and there's lots of publications on FTD. So I do think that's a model for how it might be done, although people continue to struggle with the proper asset for doing it. The large studies like that are very resource intensive. What I would suggest is more elegant experimental designs earlier so that we can work through some of the basic concepts before the huge investment, the very involved procedures that take place in a more definitive study, would occur. So FTD is a great model for that. We find that they operationalize distinctively different than we might encounter in mild to moderate AD as an example. They're very different studies, particularly if there's a behavioral variant of that disease involved. Different family dynamic, different structure, different questions one needs to ask. So earlier phase programs are more interesting in some respects to me to work out some of the uncertainties that exist in something like behavioral variant FTD. And what was the other question? I'm sorry that you
But
had?
it was, have we made any progress, I think was the main question in this area. And it sounds like there is one big trial that has made some progress.
In terms of methods, all assets have deficiencies. You don't know the dose, you don't know the duration, you're not sure about the measures. But in terms of designing studies which are informative, I think there's been tremendous progress in FTD that would enable a very informed and staged clinical development program. So we call it development to an inflection point, development to a point where you make the next decision. I think there's a terrific amount of art present internationally in that particular indication. Yeah.
Now we have a question.
Oh, I could,
Your thoughts?
the comment I would make is I know small companies are doing antibodies to progranulin as a therapy, but that in my eyes is just another example of trying to make an antibody, external antibody outside the brain that wouldn't penetrate the brain and against a particular antigen that's clearly linked, but we're not sure how it causes or is involved in the disease.
Yes, you're kind of doing discovery in development. We actually were part of a program in that particular phenotype, and it's a published article. So the way we look at these unique phenotypes is they're kind of discovery in development. You're not quite sure what to look at, how long it should be, what the dose exposure response relationship looks like, what the measures are. You're guessing at a host of different issues, which brings up the nature of doing things more efficiently, particularly in an adaptive program where you're capable of modifying the studies as you move through the development pathway. It's about the design. It's about how to create the program that's maximally informative with minimal expenditures of resources. The true expression of translational medicine is to be integrated with the discovery teams that are discovering these very interesting and clever molecules. So as creative as we are non-clinically, and indeed it is very creative, there's equally creative and fascinating technologies available within clinic to look at target engagement, to see how it translates into a clinical readout that's interesting. Is the clinical readout clinically relevant or just academically of interest and enables the next stage of the program? So all of these different subtypes are very fascinating topics. And I honestly believe that mastery, broadly speaking, in this particular therapeutic area requires mastery of methods, where you're able to translate between the non-clinical observations and clinical trial design in a team structure.
Okay, no, go ahead.
So I was going to say part of the issue is the non-clinical models don't translate. So that's a problem. And I also think the progression of disease, yes, you can do the early clinical trials and show you have target engagement across the BBB, and maybe there's some biomarker change, but then you've got to end up again crossing the finish line and showing you have clinical benefit. And one of the things I know we thought about at Novartis is, what are the neurodegenerative diseases where the progression is fast enough that in a reasonable time frame and sized power trial, you could actually show decreased progression? And so also choosing your patient population that way.
That's right. Yeah, we call that a leveraged patient sample, a patient that's somewhat idiosyncratic to the general population, and you pivot on certain neuroimaging, neurochemical assessments in order to make adaptive decisions in the program. It's a fascinatingly interesting topic and I think one that's, at least in our experience, is increasingly voiced by most of the sponsors we have the privilege to work with. How to incorporate advanced technologies into early phase research to enable the next step in the program. Yes.
Okay, so can we also, we have several questions now. Okay, so so first question, but I want to ask something Peter later on, and Jonathan, but anyway, I'm the last. So first question, a very promising approach is treating neurodegenerative conditions with red or near infrared light. What would you think about that?
Anybody know?
Okay.
I can say I've not encountered that one.
So something we all need to learn about.
Okay, other question that we have, which is from Thomas. So do you think, do you have any comments on other cardiometabolic or other routes that could be targeted upstream of inflammation?
Are you thinking, Thomas, like hyperglycemia or
Something, or is it something
or
upstream from
lipids,
inflammation?
right? Hypercholesterolemia.
Or maybe some metabolic dysregulation in the yeah, that's probably what you're trying. Yeah.
Any comments?
Well, the comorbidities broadly defined impact disease progression, and it is possible to examine selective subsets in various programs. For example, in Alzheimer's disease, it is possible to examine that hypothesis. A number of companies have actually approached that, about looking at, I call it disease modifiers in a very informed way. I do think the comorbidity question as a driver for, say, dementing illness as such is a fascinating topic, and with a wealth of basic research to support it. So in our experience, is how you design that program that really counts, and how do you extend it into more definitive studies.
I think
More
we'll
hypothesis
hear.
generation, not hypothesis generation more than testing.
I think Novo Nordisk, and we're going to hear about this, has found that their GLP-1 agonists have very promising effects on cognitive function or Alzheimer's disease in obese or diabetic patients, and they're actually doing an Alzheimer's trial with GLP-1 agonists.
There you
That
go.
should be very exciting. Again, probably nothing to do with inflammation,
Or
maybe,
maybe.
but I think that is what Thomas was getting at. Really interesting link.
So Peter, can you please elaborate a bit on the data that you showed us on what exactly are you targeting with this molecule that you are
Maybe just one step back, I also want to add. So I think that it's clear that you think mouse models are not always the best to predict the response in patients, and for understandable reasons. But I think for some aspects, especially inflammation, mice are actually quite useful, right? So we have now quite some evidence that senescence protein, SP16, can also form amyloid-like structures. And this can actually enforce further inflammation. So by dampening the aggregation of some of these senescence-associated proteins, you can actually also downregulate mitochondrial stress and the associated CGAS sting and the inflammation pathways. So you don't always have to kill senescent cells. We can also modulate how they behave. And that is actually very well, we have very good models to study this in 3D organoid cultures and also in mice. So I realize that, a model is a model, and it's definitely not a human being. But I think these aspects have been well characterized now in mice. And I think compounds to target this are warranted for clinical translation because of that. And to come back to your question on what we are targeting, our peptides are very niche-specific anti-senescence compounds. As we heard also from Paul Robbins, there are many markers of senescence: P16, P21, lemon B1, HMG1, SABTGL. There are many different ones, and that's the problem in the field. Our compounds target a DNA damage high form of senescence, which we call scarred senescence. So there's a lot of scarring ongoing in these cells. And that's hallmarked by an activated form of P53 and a second protein FOXO. And that is not in all senescent cells. So that's a DNA damage associated form of senescence, and that leads to an inflammatory response. So IL-1 alpha beta are through the roof in these cells, right? So I think it's relevant with what we heard today, also from what Ted Dawson was arguing for, and also I think what Joan and Mike were arguing for, that inflammation is a problem. Maybe not A beta or tau, I'm not going to say anything about that, but inflammation for sure is increased by these P53 high scarred senescent cells, and these we can eliminate in cells and organoids and mice, and we are now going to clinical trials for oncology. So that is actually for our company the first indication, but I'm very, very curious to see if we can employ this also to neurodegeneration in a later phase. Yeah.
Very interesting.
What about the fact that inflammation also causes vascular disease, and vascular disease also contributes to neurodegeneration?
Yes, 1989. The concept, of course, has been tremendously discussed at some length. And that's part of the problem we have, I think, in this particular area, is that the diseases are overdetermined in many ways. Or I call it overdetermined. There are many different etiologies. And so creating a testable program that identifies a certain hypothesis, allows you to test a hypothesis in a way that's informative, is really quite a challenge. So, we use the term a leveraged patient sample, patients who are somewhat idiosyncratic for the disease in question, but nevertheless either have some genotypic or phenotypic quality about them that allows you to test an hypothesis out in an interesting way. So it is an important concept you've raised.
Okay, one other question that I would like to ask Jonathan is, what do you think actually nicotine is targeting? You're not alluding that we all become smokers.
I'm sorry, did you repeat that?
What do
again?
you think actually nicotine is targeting? So how does it affect the vagus nerve? And could you elaborate a bit on your hypothesis there?
The point is the amyloid will bind as agonists of the vagus nerve and would be immunosuppressive. The question is, really, how much does the brain monitor brain inflammation? Because the vagus nerve is monitoring distal inflammation in the gut, the liver, the heart. And I do not know how the alpha-7 signaling pathway in the brain is going to affect inflammation there. So all our research led us is to the conundrum that amyloids were not universally inflammatory. In fact, that they could bind and be immunosuppressive. So we have not pursued that. We went after orally bioavailable drugs that stimulate alpha-7 for, again, indications in the heart and the gut, not in the brain.
And Ted has a comment to make. Ted said that leveraged patient samples are very challenging to identify in neurodegenerative diseases.
That is certainly true, but you can always argue that the early phase research is never representative of the ultimate sample to receive the product. But nevertheless, the eligibility criteria by definition makes it not generalizable. So the question is, how do you leverage the sample using either biochemical, imaging, other neurochemical aspects, other actually assessments that to enhance signal detection for hypothesis generation as well as testing, creating in some respects a sample which is slightly idiosyncratic from those that you eventually want to treat in order to create a testable hypothesis. This is the challenge we have, particularly in early phase research. But we clearly see an evolution in generalizability as the programs evolve. As the programs evolve, the patients become, we say, more generalizable. They're actually very discrete and very precise phenotypes, sometimes genotypes, in the earliest phases of clinical research, generating more questions, I think, than answers, so that you can enable a subsequent program. Yeah.
I was just thinking, the aducanumab and the current therapies are all seeming to cause, as a side effect, increase neuroinflammation. Are we going to end up needing combination therapy? Is it that we'll need an anti-inflammatory and maybe also an anti-protein aggregation play?
Well,
Anybody?
it's been
thoughts?
discussed for 20 years. So it's, as we all know, these diseases have many different contributing factors, many different hypotheses that all seem to have some merit. And the short answer is probably it's a cocktail at some level in
Right.
terms of how we treat these different disease states. But the early phase clinical research is key to enable the hypothesis. It's key to actually generating the unique patient phenotypes, maybe a cocktail approach to therapy in some cases. We recently had something like that in ALS as an example. So valid point, well taken.
And where, if for each of the panelists, where do you think, which neurodegenerative disease do you think is going to be the one that we're going to crack first and really make meaningful strides in stopping, reversing, preventing? Ted, I'll start with you.
Ted, yeah, well, he said,
We're starting going around a circle.
Well, it's a daunting question. I would, because I'm closest to Parkinson's disease, I would like to think that there are really a lot of promising therapies in the pipeline. It has many of the challenges that have been mentioned with doing clinical studies, but they're well-defined, criteria for neuroprotection in Parkinson's disease. And if there is a compound that can get over that hurdle, I think it's going to be the first one in the many diseases. We just have to wait and see.
One of the fascinating things from my minimal experience in Parkinson's trials is no one could tell me, and I guess people just don't know, is it better to increase alpha-synuclein or decrease alpha-synuclein levels in the CSF? And that always worried me. Like, that's a tricky biomarker. But you're right. I think movement measurements are maybe a little bit easier than cognitive function measurements. And so we may have an easier regulatory clinical path for Parkinson's disease. So Jonathan, go ahead.
I'd rather do a comparison. See, Parkinson has a very nice runway. You have 10 years often with these patients. Compare that to ALS, right? ALS is extremely acute. So that the practical aspects of modulating Parkinson's is certainly an opportunity. Alzheimer's, I think we all agree, is multiple diseases. The early onset is a completely different indication than the late onset. And the late onset is fraught with elderly people, half of whom do not survive your clinical trial. So I think Alzheimer's has a fundamental problem, except it's the largest market. And large pharma has their focus on market size. And large pharma, to their credit, is the one's going to crack many of these cases. Huntington's, I know less about than Parkinson's. But I can't exaggerate. There might be breakthroughs in schizophrenia before there is in some of these others, and then that could be switched over, because synaptic pruning is clearly going to be an issue, or death of any of the synapse with the microglia is going to be fundamental to cognition.
What about MS? What do you think about MS? I know it's not
We're
it's not the
already-
clear
right.
it's not the clear
And we have
neurodegenerative.
therapies for it.
Ah.
Well,
Jonathan?
certainly we're moving toward more refined phenotypes across the board. You know, I recall a time when we had organic brain syndrome as a target. So now we're talking about very refined phenotypes, probably phenotypic genetic interplay that eventually characterize the patient population we're treating. You know, what we called Alzheimer's disease 20 years ago is not, that's for sure. And that goes for any of the other neurodegenerative conditions we might speak to. So I think more refined phenotype, a combination of phenotypic and genetic information to fractionate the population, more mosaic of measures, clinical measures, obviously, appropriate to stage of disease, but also neuroimaging, neurochemical, electrophysiological, a collage of outcomes rather than one outcome, dictating whether there's a go-ahead on a given product.
Absolutely. And Peter?
We had a very large conference last week, or offline, in the senescence field. And if I deduct from that, what I think will be the first is it is tauopathy related. So whether that's Alzheimer's or not, there are some amazing studies that are about to be sent out for publication, because they were now presented at the meeting. And there are now things called eigengenes. So there's subtypes of senescence markers in tauopathies. And people have been making compounds and getting successes in mice. So I'm sure that these companies will be looking for clinical translation soon. So I would have to go with tauopathy-related Alzheimer's disease. And then also, this is related to the later Brach stages. So that, of course, touches upon what Jonathan says, that maybe these are beyond, saving, if you will. But I think at least what I've seen in these mouse models, you can stall the progression of the disease with these compounds.
Right. I totally agree. I think the progression is going to be delay of the progression of the disease. And if you give these people 5 more years, it's going to be very much like the cancer models. If you can give improved quality of life for a period of time, that's clearly what some of these new ideas are going to result in.
Yeah, one of
rather than cures.
Yeah, thank you, Jonathan. I think one of our challenges is that the treatment effect might be asymmetrical. It might be only in one domain, but not all domains. And so the ability to parse out that signal in what usually is a longitudinal clinical trial is the art of doing this, particularly early stage research. It's not a uniform construct, both in terms of diagnostics, as well as in terms of assessments that one might use. So kind of a differential weighting of outcome is direction we're kind of going in, where you look at the totality of data, particularly in early phase research, to provide a go-ahead signal for these diseases.
Yeah, to that point, Peter, I love the concept of diminishing the senescent cells, but it's almost like weeds in a lawn, more are going to come back. So there's an example of an idea or a concept that would certainly lead to progress without cure.
Yeah, that's a good point. So, like I said, it's this trade-off between more buildup of DNA damage throughout life, and later phase in life, it goes faster and faster and faster,
Right.
versus declined immune-based clearance of senescent cells. So, indeed, you're fighting against the inevitable in a way, but
right.
That said, you can remove these cells at regular intervals, right? People have done that. So it's so far, chances of resistance towards intermittent clearance of senescence has not been observed. So you can remove these cells, let's say, every 3 months, or well, in mice at least. I mean, and hopefully in humans, maybe every once a year. We have to figure that out. But it's not like that's why I was pushing back a little bit towards chronic treatment with anti-inflammatory drugs, where I fear, but that may be unbiased fear, that you will get resistance at some point. But that's, again, it's up for discussion. So I think a TNF-alpha approach is definitely worth considering. Yeah.
And I meant to bring up both. Peter and Ted have been talking about a lot of DNA damage occurring. I guess it's mainly in the microglia and the astrocytes in the brain. Did I get that right?
Mhm.
We
Ted,
have
was
seen
that?
senescence in neurons as well, but it's uncommon. Yeah.
So
Well, you can see, I think with regards to senescence, it's occurring in the microglia. When DNA damage occurs in neurons, the neurons die for the most part.
Yeah, exactly. That's it. So we do see that if you have microglia that's senesced, then the neurons contract. So you see a consequence of annoying microglia on the neurons. But
And how about the astrocytes? Do they have DNA damage?
They do, but it's at least in our hand, it's been harder to detect. I think the main cell population of this is microglia, but
So interesting.
Can you, for the lay people out here, that not the non-specialists, sort of wrap up? What are we to take away from sort of these inflammation and other approaches to neurodegeneration? What should we expect?
Well, my experience has been there's a lot of ways around the backside of space in this indication, and that there are many complementary hypotheses that warrant an explanation. It's often been said that a cocktail approach might ultimately be the intervention required. The one thing I would take away, given our limited time, having done this for many years, is that this type of initiative requires a collaborative effort amongst different individuals, each of whom bring a different perspective to the table. That's the one takeaway I've had doing this since many years ago, because no one of us alone is going to be as good as all of us together thinking through all these different constructs. So,
Well, thank all you panelists, our experts. Thank you so much.